Microgametogenesis, abnormalities, that is the anemic phenomenon. A male gamete or a pollen grain was usually 2 or 3 nucleated, but under abnormal condition, dominance of female potency was observed, where a male gamete was 8 nucleate, showing a surprising resemblance to an embryo sac in the female gamete phyte. It was first observed in anthers of flowering plant species named Hyacinthus orientalis by botanist Nemec in 1989, and he called it as pollen embryo sac. He believed that pollen embryo sac arose as a result of degeneration of generative nucleus and 3 divisions of vegetative nucleus of the pollen. De Mol in 1923 observed this so-called Nemec phenomenon in anthers of other varieties of Hyacinthus orientalis, which had been subjected to certain special conditions in order to obtain early flowering. He attributed the origin of abnormality to a duplication of generative nucleus. Kettler in 1941 and Ram in 1959 reported such structures in Ornithogallum nutans and Leptomeria respectively. These findings indicate that pollen embryo sacs are formed by 4 ways, that is microspore mother cells, microspore nucleus, vegetative nucleus, generative nucleus. Stowe found similar embryo sac-like pollen grains or pollen embryo sacs in the anthers of a variety called La Victor, whose bulbs had been subject to a temperature of 20 degrees centigrade at the time of meiosis and were further forced in a greenhouse. He traced their development more fully than either Nemec or De Mol. At first, the microspores increase in size to form a large sac-like bodies, after which the nucleus undergoes 3 successive divisions to form 8 nuclei. Of these, 3 lie at the end where the exine is still intact, and 3 at the opposite end and 2 in the middle. The 6 nuclei at 2 poles organize into a cell, while the remaining 2 in the center fuse. Since the 3 cells at the exine end were found to remain healthy for a much longer time than those at the opposite end, Stowe regards the former as corresponding to the egg and synergids and the latter to the antipodals. Abnormal pollen embryo sacs. In addition, certain abnormal pollen embryo sacs were also seen, showing the following type of organization. That is 8 nuclei forming an egg, 2 polars and 5 antipodal cells. The second one, 4 nuclei forming an egg, 2 polars and 1 antipodal cell. The third is 4 nuclei forming a polar and 3 antipodal cells but no egg. The fourth type, 16 nuclei forming a 5 to 10 egg apparatus. In conclusion, Stowe says that all the pollen grains are potentially capable of assuming either the male or female form. Under normal conditions, the male potency is dominant over the female potency, leading to the formation of generative cell and male gametes. Under abnormal conditions, when there is a release of necrohormones, a hormone which causes abnormal growth of surviving pollens, the female potency gets the upper hand, resulting in formation of embryo sac-like structures. So, the summary of this is Nemec observed embryo-like pollen grains in the petaloid anthers of Hyacinthus orientalis. He believed that they were formed by 3 successive divisions of vegetative nucleus. Later, De Mol and Stowe noted that Nemec phenomenon in some varieties of Hyacinthus orientalis, which were subjected to special treatments. Subsequent, Gaitler and Ram reported such structures in Ornithogallum nutans and Leptomeria. These findings indicate that pollen embryos sacs are formed by 4 ways: by division of microspore mother cell, microspore nucleus, vegetative nucleus, and the generative nucleus. Under normal condition, the male potency is dominant over female potency, leading to the formation of microgametophyte. Under abnormal conditions, when there is a release of necrohormone, leads to the formation of pollen embryo sac. Thank you.